everyone, the study that we'll be presenting today is entitled The Role of Dopamine in Modulating Intermale Courtship Behavior in Ethanol-Exposed Drosophila.
We are studying Drosophila, and it's not uncommon for a male Drosophila to exhibit intermale courtship behaviors. They are one of very few species to do so, and there have been experiments in the past that have been conducted that have been trying to pinpoint which neural systems or genes are responsible for modulating this behavior. Previous research has shown that manipulating dopaminergic neurons and dopamine receptor expressing neurons is correlated with increased instances of intermale courtship behaviors. This likelihood that male Drosophila will display typical courtship behaviors towards other males is measured by a courtship index, which we will discuss in more details later, and it increases when dopamine production was both increased and decreased. Manipulating dopamine receptors also had the same effect. Additionally, administering ethanol of increasing concentrations also increases the likelihood of intermale courtship behaviors. However, we have not seen any existing literature that implements optogenetic techniques to manipulate the dopaminergic neurons and dopamine receptor expressing neurons. The previous research experiments did not introduce channelrhodopsin or halorhodopsin into neurons. Instead, they used temperature-sensitive alleles and other techniques that changed the neural anatomy of their mutant flies. To the best of our knowledge, there were no experiments conducted in which dopaminergic neurons and dopamine receptor neurons were stimulated or inhibited in real time. And by real time, we mean as the flies are interacting with each other. Also, there was no previous studies before that we could find that manipulated a dopaminergic system in conjunction with ethanol administration. Manipulations to the dopaminergic system and ethanol administration experiments were always conducted separately.
According to the NIH, approximately 14.1 million adults have alcohol use disorder. Alcohol is largely implicated in modulating social behaviors and commonly increases the tendency to engage in normal and risky sexual behavior, such as unprotected sex. Addiction studies have suggested that dopamine plays a role in alcohol abuse, potentially by altering the mesolimbic dopamine systems of alcohol-dependent people. But this role is complicated and requires further study. Drosophila are a model organism for studying social behavior, and they share approximately 75% of disease-encoding genes with humans. This high genetic similarity makes them an ideal candidate for studying manipulations to neurobiological systems and relevant consequences for humans. Therefore, our experiment is important to further understanding the role of dopamine in disinhibiting sexual behavior during intoxication. The main question that we proposed and explored through this experiment was how the activation or inhibition of dopamine with optogenetic manipulation in ethanol-exposed flies impacts intermale courtship. So to answer this question, we pursued three specific aims. The first being genetically mapping and visualizing the dopaminergic system. The second one being examining the effect of optogenetic manipulation of dopamine neurotransmission on intersexual courtship. And the last one being examining the behavioral consequences of manipulating dopamine D1 receptor activity.
For our genetic crosses, we used the GAL4 UAS system, and the first cross we created was between a UAS line expressing redshifted channelrhodopsin and a GAL4 line targeting dopamine neurons. And for our second cross, we also expressed the same redshifted channelrhodopsin from the same UAS line, but instead of neurons, it was expressed in D1 dopamine receptor expressing neurons from a GAL4 line. And for our experimental design, which the setup can be seen here, we had varying levels of ethanol placed in a filter flask, and that sat on a hot plate set to 135 degrees Fahrenheit. And a tube providing airflow was placed through the top of the flask, while another tube was attached to the side of the flask to connect it to the main chamber, or the plastic box that you can see. And within the plastic box, the flies were placed in a plastic vial that had a mesh lid to allow the entry of the ethanol. And on the opposite end of the chamber, another tube exited, and that was attached to a vacuum. and this air and vacuum system just ensured the distribution of ethanol throughout the chamber.
After each pair for each trial spent 10 minutes in the chamber together, the vial containing the flies was removed and the trial officially started. Each trial was 5 minutes long, broken up into minute intervals that alternated between the red light being turned on and being off, starting with the light off. Over the 5 minutes, the interactions between the two flies were recorded using an iPhone camera, and then after the footage was captured, it was uploaded, and each group member individually watched the recording and was able to score off of the behaviors listed above for each minute interval. So we were looking for behaviors such as orienting, like facing each other, tapping, singing, which was basically just their wings vibrating, licking, attempted copulation, and we also counted following as a courtship behavior, which is not shown above. And then after each member had the chance to review the footage and score how much of each minute was spent of the flies exhibiting these behaviors, all of the recorded measures from each member were averaged, and those are what we use for our behavioral assay, which we will show later. We got this behavioral assay from Liu et al. This is what he did in his study. So then before we actually got to the behavioral assays, we were able to capture these images that are visualizing the Drosophila brain using fluorescence microscopy. The image on the left shows the Drosophila brain, and the cells are labeled with green fluorescent protein, GFP, and on the right, they're labeled with DAPI, which normally is supposed to look blue, but ours turned out a little pink, reddish, because there was some of the eye film, which is red, that got dragged in with the brain while we were imaging. And then here are the graphs displaying the averages of our behavioral assays. There were 3 trials for each line, the dopaminergic and the dopamine receptor expressing neurons. And each 3 trial was exposed to varying amounts of ethanol. The first of the 3 trials for each line had no ethanol exposure. The second one had 35% ethanol, and the last had 70%. For the dopaminergic neurons that were not exposed to ethanol, we used deionized water. We also use it for the receptors, but and our results are shown here. There was not much of a difference between the minutes where they were exposed to the light and when they were not. There was pretty steady activity across both situations, but for the 35% ethanol trials, there was activity focused mainly in the middle of the trial during the second and fourth minute, between the second and fourth minutes. And since during the third trial, we did get a bit of activity, but the light was not on, we can't conclude that the increase in behavior is a result of the active exposure to the light. For the 70% ethanol trial, most of the activity occurred in the first minute, and then locomotion in general decreased throughout the rest of the 5-minute period. Presumably, this is due to the effects of exposure to the ethanol.
So for the dopamine receptors, across all 3 conditions, we didn't see any differences, or rather any mating behavior. And although this isn't extremely exciting, it's also what we expected. And this is because in this condition, we weren't introducing any new dopamine, rather we were just stimulating the dopamine receptors. So it is expected to not see any increase in courtship behaviors. However, this does lead to the question of the impact of ethanol, as previous research has shown that ethanol independently tends to increase courtship behaviors. And since we didn't see that, that is something that is a little bit out of the ordinary and could potentially be researched in future research.
We didn't see any statistically significant results, but with further experimentation in a larger lab setting, we would presumably be running this experiment with many more trials. In future studies, as Petal mentioned, it will be beneficial to isolate ethanol on its own because our results didn't align with previous research, which showed that increasing ethanol dosages to Drosophila correlates to increased instances of intermale courtship, which we did not see. If we continue running trials like this, we will be able to better understand if the reason our results don't match up with previous experiments is because we added the second variable of dopamine manipulation, which may have negated the effects ethanol should have had, or if there's another reason for this misalignment. In addition, we only introduced channel rhodopsin into dopaminergic neurons and dopamine receptor expressing neurons, so we only got to test what would happen if these neurons were stimulated. Because previous research has also shown a link between inhibition of these neurons, which would also increase intermale courtship, in future experiments, it would be interesting to also apply optogenetics to inhibit dopamine neurons as well, using the introduction of hyaluronic. Thank you.